Hi guys, in this series of videos we're going to be looking at what exactly enzymes are, how enzyme function relates to structure and how enzymes work, and we'll finish with a summary.
So enzymes are biological catalysts. The reason they are called biological catalysts is because they speed up metabolic reactions. Now, catalysts are just generally chemicals that speed up rates of reactions, and metabolic reactions are any chemical reactions that take place in a living thing. So that's why we call them biological catalysts. Because they're catalysts, enzymes are not themselves used up in the reaction, which means that they can just catalyze a reaction and then go off and do something else. They're essentially recycled, and because they are recycled, just a small amount of catalyst can actually catalyze a reaction involving lots and lots of molecules, because each one can be used again and again. And in fact, the turnover number of an enzyme describes the number of reactions one enzyme molecule can catalyze per second, and they can get pretty big, as we will see later. So why are enzymes so remarkable? They can be compared to chemical catalysts. Biological catalysts are remarkable mainly because they work at normal physiological temperatures, whereas chemical catalysts need very high temperatures often to work. In addition to this, they also need very high pressures and can need extremes of pH. Chemical catalysts are also less specific. Enzymes don't tend to make any unwanted byproducts, and this is because they rarely make mistakes. So what I mean by this is that the products from enzyme reactions are never going to be contaminated with stuff that's come from those particular chemical reactions going a little bit wrong. Biological catalysts or enzymes are also regulated by cells, so you don't need a host of scientists to help make life work. It's a self-regulating process. You can roughly split up enzymes into two types. You have intracellular enzymes and extracellular enzymes. Intracellular enzymes are the ones that will always be involved in metabolic pathways. Within any cell and within its organelles, there may be up to 1,000 metabolic reactions going on at any same time, and each is catalyzed by a different enzyme. Now, these reactions might be part of metabolic pathways, and I've drawn out a rough sketch of one here. So the letters in red show the enzymes, the numbers would be the substrates or reactants and products and intermediates, and the green arrows show an example of how regulation might happen. So this, as you can see, is already relatively complex, even though it doesn't actually describe a real metabolic pathway, but it's quite representative of what they end up looking like. They look more like networks than actual pathways. So each metabolic pathway is a series of consecutive reactions. For example, 1 is turned into 2 by enzyme A. The various reactants and intermediates and products of the reactions are known as metabolites. And in some metabolic pathways that split things up, i.e. substrates are broken down into smaller molecules and releasing energy, these are described as catabolic reactions. Anabolic reactions are the opposite, where two reactants are brought together to form one product. Now, we also have extracellular enzymes, and these kinds of enzymes are usually secreted, and they're usually used for digestion of molecules before you uptake them. So a good example of this is fungi. Fungi have these things called hyphae, which is basically this long filamentous, almost thread-like network of fibres, and hyphae are actually used to secrete hydrolytic enzymes, for example in bread mould, which helps the fungus to digest the thing that it's living off before it actually absorbs the molecules. Now, we've got some important examples of both of these kinds of enzymes. So for intracellular enzymes, it's important to know about catalase. So catalase catalyzes this reaction, which is 2 lots of hydrogen peroxide, or H2O2, which goes to oxygen, and 2 lots of water. Now, hydrogen peroxide, as you might be able to tell by its effects in bleach, is quite a dangerous product, and it's actually a byproduct that's made in lots of metabolic reactions. So obviously cells want to get rid of it, so catalase is a very important enzyme which allows them to do this and convert hydrogen peroxide into harmless oxygen and water. Catalase consists of 4 polypeptide chains and 1 heme group, which contains an iron. Now, its turnover number is large. It is 6 million reactions per second. And this is actually the highest turnover number known for an enzyme. In eukaryotic cells, catalase is often held in peroxisomes, which are just small vesicles containing catalase, and white blood cells actually use catalase to help phagocytose or digest unwanted pathogens or organisms before they are gotten rid of and disposed by the rest of the immune system. For extracellular enzymes, we have a couple of digestive ones. So we have amylase and trypsin. Amylase is secreted by your salivary glands, and it catalyzes the reaction of starch to maltose. Starch is a polysaccharide, and maltose is a disaccharide. If you're unsure of what those mean, I would recommend checking out the carbohydrates videos. Now, amylase is also secreted by the pancreas into the small intestine, where it also helps to catalyze this reaction. And trypsin, like amylase, although it's not found in your mouth, is also secreted by the pancreas and works in the small intestine to break down proteins into smaller peptides. Now, as enzymes are very specific and take part in so many important reactions, obviously having enzymes that are dysfunctional can lead to certain deficiencies. However, how are enzymes exactly so specific? We will see how in the next video, having now looked at some examples of enzymes and some of their properties.